All right, everyone. So let's go ahead and continue here talking about cancer, basically. So we've already covered this in class, and we talked about how cancer is basically a bunch of different diseases, but they all have the same underlying mechanism. Okay. So even though they all may occur in different places in the body or things like that, ultimately the main cause or the main mechanism for cancer is just uncontrolled cell division, okay? And that is because, well, even though our cells have this amazing ability to divide and just make more of themselves, that needs to be controlled, okay? Once they go rogue and they start going about their own business without listening to the signals of the body, that is when we run into issues, okay? So let's go ahead and talk about how we actually regulate the cell cycle, okay? And that happens through a series of checkpoints. The three most important ones are highlighted in here. We have a checkpoint here during G1 of interphase. We have another checkpoint right here, also during interphase, but in this case is in G2. And then we have one last checkpoint that we need to know about or that we care about at this point, and it happens during the actual phase of mitosis, okay, the actual division of the nucleus, okay? So let's talk about a couple of things that are gonna come into play here, right? So one thing that our bodies use to regulate the cell cycle itself is basically fluctuations of these proteins, these signal proteins called cyclins, okay, cyclins. And they are going to basically come together with these other types of complexes called cyclin dependent kinases. Okay, here's that name. It's very long. Cyclin dependent kinases, or kinase, okay, for short. So as with anything, with any new name, I always encourage you to try to say it yourselves, okay? So if you're listening to this recording, go ahead and pause it and try to say the name. I know it's hard, but that's one good way to start learning some of this new vocabulary, okay? So we're gonna have cyclins and we have cyclin-dependent kinases, okay? We're gonna call them CDKs for short, all right? So cyclins are these proteins, these again, signal proteins that are going to fluctuate in concentration, okay? Their concentration is gonna go up and down throughout the life of the cell, all right? And whenever that concentration goes up, they will actually form a complex with the CDK, all right? And that is why they're called cyclin-dependent kinases, because, well, these enzymes do not work unless they are coupled with a cyclin, okay? So the CDKs don't really change in their concentration. They're always in the same concentration in the cell. However, whenever it is not attached to a cyclin, then the kinases are basically inactive. They're just not doing what they need to do. Once the cyclin concentration goes up, then they form a complex and they start activating the cell cycle. The way that they activate the cell cycle is by some phosphorylation. They're going to be phosphorylating some enzymes and some molecules that are going to get the cycle going. And this specifically happens at this point right here. That is that G2 checkpoint that we were talking about, okay? So whenever we have this cyclin CDK complex together, which in this case is the maturation promoting factor, or MPF, that is basically going to tell the cell, yes, you are ready to divide, okay? So up until we have that complex formed, which is right here, okay? It's signaled right here. We have this MPF, okay? The MPF is putting together a CDK with the cyclin that all of a sudden increase in concentration. They're gonna come together and that tells the cell, all right, you're good to go. Go ahead and divide. Up until that happens, the cell is going to stay in G2. It is not going to move forward, okay? So once it go ahead, the cell divides, then that whole complex is going to break apart. The cyclin is going to be degraded, and now the CDK is going to be by itself. When the CDK is by itself, it's not really going to do anything, okay? Because again, it depends on the cyclins. So the only other time that it's going to start working again is when a cyclin accumulates, goes up in concentration, and then it forms a complex again. So that is what is guiding the cell cycle itself, is that coupling of the CDK with the cyclin, okay? And that basically comes into play in the G2 checkpoint. Now we have other things that control when the cell divides or when the cell should stop dividing, okay? So one of the things are called growth factors. Growth factors are released by some cells, and basically they're going to tell surrounding cells or other specific cells that, hey, you guys need to divide too. So it's basically to stimulate other cells to divide. And that is what happens with our immune system quite a bit. So whenever there's an infection or something like that, or you get a cut, so at that point, you're going to need some of your immune systems to make more of themselves so that they can either take care of that infection or close that wound, stop the bleeding and things like that. So those cells are going to tell the cells around it, hey, divide, make more of you because we have something to take care of here. And those are called growth factors. That is what is being released to notify the cells to make more, okay? We also have density-dependent inhibition. So in this case, it's kind of doing the opposite. Whenever we're getting too many crowded cells, they're going to start sending signals to tell the other ones to just stop dividing because, hey, there's too many of us right now. We don't need to divide anymore for now, okay? So it's actually doing the opposite. Whereas growth factors are telling cells to divide, density-dependent inhibition is telling the cells to stop dividing because it's no longer necessary, okay? Anchorage dependence, okay? Anchorage dependence, that's basically something that happens with animal cells for the most part. They need to be attached to something to divide. They're not just going to divide while floating in the middle of nowhere, okay? They need to be attached to something specific for them to know, hey, I'm in the right place. Now I can divide. If they're not attached to the right conditions or in the right area, then they're not going to do cell division. And that is another way that we control when and how the cells divide within the body, okay? Now, so here's a summary of all of those checkpoints, okay? We talked about the G2 checkpoint right here. That happened with those cyclins, okay? But let's go ahead and go back here to G1, all right? That is that first checkpoint, and it's more like a green light, red light type situation. So as we talked about when looking at the cell cycle, when cells are in G1, they're just going about their business and performing their functions, right? They're growing, they are doing what they need to do depending on the type of cell, whether it's a brain cell or a liver cell or anything like that. They're just going about their business and doing their functions and doing their metabolism, things like that, okay? And for the most part, they're going to stay there, okay? Because at this point, they're going to have a red light that tells them, hey, no, you don't need to divide right now. So stay where you are and do what you need to do, okay? And some cells even completely exit the cell cycle and go into G0. G0 is called cell cycle arrest, okay? So that means that this cell is never going to divide again. Examples of cells that do that include brain cells. Our brain cells do not divide, okay? They are in this state, G0, called cell cycle arrest, okay? Now, for any other cell that will get that green light over here, it's basically telling the cell, okay, time to move on to the next portion, which is what? What comes after G1? Well, that's going to be the S phase, right? That synthesis phase where the DNA is going to replicate. So once the DNA replicates, the cells are going to enter G2. Okay. Now in G2, the cell is going to stay there up until we get that cyclin CDK complex that gives the cells the go ahead to proceed with cell division, okay, or mitosis at this point. So once it goes into mitosis, the last checkpoint is going to be right here during mitosis. So actually, I think I have these things going over here, right? So the M checkpoint, it's basically going to tell the cell that it cannot finish division up until all of the mitotic spindles are attached to the chromosomes, or that at least I should say it's all the chromosomes are attached to the mitotic spindle. That is because if it doesn't do that, then it may end up separating the chromosomes in the wrong way. And some cells may end up with more chromosomes than necessary, and the other cells may do the exact opposite. Okay. So those are all of the things that our cells have to ensure that they are dividing in the right way and dividing only when it actually needs to happen. But when the cells start ignoring those signals, okay, when the cell just completely disregards what is happening around it, we call that transformation, because those cells are now going to have the ability to divide indefinitely. Those are cancer cells, okay? They do not pay attention to the cycle, okay? They don't pay attention to the CDK cycling complexes. They don't stop dividing even when there's no growth factors. And also something that happens is that they basically stop repairing their DNA. So we talked about DNA damage. We talked about DNA repair last week. Cancer cells lose that ability. They don't care anymore. They're just making more and more and more of themselves, okay? So I'm sure you guys have heard about benign tumors versus malignant tumors. Benign are those that stay at their original site. At least they are easy to treat, or easier, because they're just going to stay in the one place. So all we need to do is remove it, okay? Malignant tumors, however, have the ability to detach from wherever they are and travel using the bloodstream to other areas of the body. That is called metastasis. Okay, that's when we spread the cancer to other places of the body and they start growing other tumors. So anybody that knows anybody that is going through some sort of cancer treatment, they're probably using some form of chemotherapy, because this is for now the most widely used treatment and the most effective that we have at this point, okay? All of the chemotherapy drugs, they are using these chemicals that target the cell cycle. And the way that they do that is that they specifically target cells that divide rapidly, okay? And this is why we have some of the side effects that we normally see in people undergoing chemotherapy. Of course, we know that people undergoing chemotherapy are losing hair, okay? And that is because hair follicles divide quickly, okay? They also become immunocompromised. And that's because the cells of the bone marrow, okay, which are the cells that are making your blood cells and your white blood cells, they divide quickly too. And so chemotherapy drugs are also going to end up harming them. That is why people undergoing chemotherapy are immunocompromised and therefore more prone to getting infections. They can also become anemic. Anemic meaning that they don't have enough blood. So they may end up needing transfusions to help them deal with this blood loss, okay? And also epithelial cells. These are the cells that are lining up your stomach and your intestines and your mouth, okay? These cells undergo a lot of stress during your lifetime, and so they are constantly being replaced. Okay, so just think about the stomach, right? Think about the conditions inside your stomach, right? It's really acidic in there. And so the cells that are lining up your stomach constantly need to be replaced by new ones. And so whenever you're undergoing chemotherapy, those cells are going to be affected. And that is why people undergoing chemotherapy treatment, they end up having a lot of gastrointestinal issues, like really, really bad gastrointestinal issues. So that is all we covered in class regarding cancer. I will see you guys on the next recording when I go over meiosis with you. See you in the next one.